Hello, my dear students of class 12. How are you? I hope you are all well and healthy. This is Morshida Mahbuba, Senior Lecturer, Department of Biology, Milestone College. Welcome you all to our online biology class. My dear students, today we will discuss about two creative questions from chapter 10. You have to maintain some tricks. You should observe the stem carefully and answer to the point. Many of you come up with the wrong answer without being able to understand the stimulus properly. You write down properly what you understand, but the stem wants different things. We can show you many examples of this problem. Suppose question, what are the classification of chromosome according to the position of centromere? The questions ask for classification of chromosome based on the position of centromere, and many students write classification of chromosome based on the number of centromere. As a result, we can give you nothing but double zero. So my dear students, read the stimulus over and over again and try to understand what the stem want, then answer to the point. Then number 2, utilize your time properly. Suppose you can keep 1 or 2 minutes for knowledge based question, then maximum 3 to 4 minutes for realization or comprehensive type question, and 10 minutes for application based question, and 15 minutes for higher skills. Total 30 minutes for a creative question. And number 3, revision and correction. You have to revise your answer script properly and make correction if needed. If you maintain these tricks, I hope you can do well and get full marks. We recently finished chapter 10. I believe you all remember it very well. However, we will try to remember again the details of the matter. We provide you some knowledge-based questions, some comprehensive type questions. Today we want to discuss these two creative questions. Creative question number 1. Describe the process of creation X of the stem. Development of pollen grain. The process of formation of pollen grain within the pollen sacs of the anther of Androecium is called pollenogenesis or microsporogenesis. The anther has 4 pollen sacs. Within the anther, 4 groups of fertile sporogenous tissue develop. Sporogenous cell divides by meiosis to form 4 haploid pollen grain. The cluster of 4 microspores is called a tetrad or tetraspores. Structure of pollen grain or microspores is your comprehensive type question for 2 marks. Pollen grain may be oval, rounded, or triangular in shape. Each pollen grain is an unicellular structure with a haploid nucleus. Each pollen grain covered with 2 layers. The outer layer is called the exile, and it is tough, it is resistant, it is thick and made up of sporopollenin. The exile is often elaborately sculptured. The inner layer is called intine. It is thin and made primarily of cellulose. The intine lies just below the exile. The exile does not develop over certain regions that define the germ pore. Okay, my dear students, then development of male gametophyte. The pollen grain is the first cell of male gametophyte. The germination registers within the anther. At first, haploid nucleus of pollen grain divides into two nucleus, one large tube nucleus and a small generative nucleus. At this condition, the anther bursts out and liberates the binucleated pollen grains. Pollination takes place at these two nucleated stages. The further development of the male gametophyte takes place upon a stigma of the carpel. Pollen grain expand by absorbing the liquid from the moist surface of stigma. The intine comes out in the form of pollen tube from germ pores. The generative nucleus divides mitotically to form two male gametes called sperm. The male gametes are non-motile and amoeboid. They are slightly unequal in size. Such a pollen will be called three-celled pollen or mature male gametophyte. The function of pollen tube is to carry sperm in the pollen tube. This pollen grain with pollen tube nucleus and sperms is called male gametophyte. My dear students, we have to draw this ovule. You have to realize the portion's name of this ovule. This is the hilum, and opposite the hilum this is caloja, and the main body of the ovule is the nucellus. Okay, my dear students, development of female gametophyte. You have to memorize the development of female gametophyte. Most of the angiosperms, 75% angiosperms, develops gametophyte through monosporic process. Single megaspores participate in gametophyte formation. Single megaspore is the first cell of female gametophyte. During development of female gametophyte from a megaspore, the megaspore enlarges and its nucleus divides mitotically into two daughter ones. Each daughter nucleus separate from other and reached each pole of the cell. Each nucleus then undergoes two successive mitosis divisions, forming four nuclei at each pole of the cell. Thus, 8 nucleated gametophyte is formed. At the same time, the megaspore enlarges into an oval-shaped structure called embryo sac. One nucleus from each pole moves to the central position of the embryo sac and fuse together to form diploid secondary nucleus. 3 nuclei are towards the micropylar end and form the egg apparatus with a central egg cell surrounded by 2 synergids. 3 nuclei are at the caloja and form the antipodals. A fully developed embryo sac with egg cells, synergids, secondary nucleus and antipodals called the female gametophyte. Definition of fertilization: union of male gamete with the female gamete in sexual reproduction is known as fertilization. Fertilization is a complex and continuous process. Fertilization in flowering plants was discovered by Strasburger in 1884. Mechanism of fertilization in plant. During pollination, the pollen grain comes to the stigma. The pollen grain has 2 layers. The outer layer is called exile and the inner layer is called intine. At some places, exine is thin. These areas are known as germ pores, through which the pollen grains comes out. Pollen grain has 2 nucleus. The smaller nucleus is called generative nucleus and the larger nucleus is called tube nucleus. Generally, this binucleated condition pollination takes place. Generative cell, which will divide to form two male gametes, and the tube nucleus makes the pollen tube. The pollen tube gradually grows down through the style and enters into the embryo sac of the ovule through the microbile. If the pollen tube enters into the embryo sac through the micropyle, then this process is called porogamy. And if the pollen tube enters into the embryo sac through chalaza, then this process is called chalazogamy. And if the pollen tube enters into the embryo sac through integument, then this process is called mesogamy. The embryo sac has 3 cells towards chalaza. These cells are known as antipodals. There are 2 nuclei in the middle called the polar nuclei. 3 cells are towards the micropyle. The middle larger cell is known as egg and the small cells are called the synergids. The egg and the 2 synergids would form the egg apparatus. The pollen tube penetrates the embryo sac between the egg and the synergid. The tip of the pollen tube brushes to liberate the 2 gametes near the egg cell. The first gamete fuses with the egg to form diploid zygote. This process is called fertilization. The second male gamete fuses with the 2 polar nuclei to form endosperm nucleus. The fusion of 3 nuclei is called triple fusion. The process of fertilization and triple fusion together is called double fertilization. Okay, description of fertilization process. The pollen grain germinates after the carpel is pollinated. The pollen grain enlarges by absorbing moisture from the anther. It ruptures and in time grows through the germ pore into a tube called pollen tube. The pollen tube gradually grows down through the style and enters into the embryo sac through the micropyle. In some plants, pollen tube enters to the egg through chalaza. By this time, the generative nucleus divides into 2 male gametes by mitosis cell division. Then the apex of pollen tube ruptures, releasing male gamete to the embryo sac. One of the male gamete fertilized with the egg is known as fertilization. The other male gamete fuses with the secondary nuclei. This type of fusion is called triple fusion. Fusion of male gamete with the egg is called fertilization. At the same time, the fusion of other male gamete with secondary nucleus is known as double fertilization. Okay, difference between fertilization and double fertilization. Union of male gamete with female gamete is fertilization, and one male gamete fuses with female gamete to form zygote, and at the same time another male gamete fuses with secondary nucleus is called double fertilization. Through this process zygote is developed, and through this process endosperm is developed. It occurs in all angiosperms and gymnosperms. It occurs only in all angiosperms. It doesn't occur in gymnosperm. Results of fertilization. The fertilized egg or zygote is the first cell of sporophyte. This zygote makes a thick wall around itself and remains dormant for a period of time. At the end of dormant period, by the mitotic cell division, an embryo is formed. After fertilization, ovule undergoes cell division and gradually develops into seed. The integument of the ovule are converted into 2 seed coat, outer coat of seed is termed as testa and inner seed coat is termed as tegment. After triple fusion, the endosperm nucleus is produced. This endosperm nucleus also goes through a series of division to form a mass of endosperm cell. It provides nutrition of the embryo in the form of starch, though it can also contain oils and proteins. In most angiosperms, nucellus tissue of seed absorbed by embryo, but in some cases this tissue remain as perisperm outer the endosperm. In most of the plant, the antipodals and synergids disintegrated, ovule stalk transformed into seed stalk. In some cases, the ovule stalk evolved as a specialized soggy extra outgrowth that partly or completely covers the seed. This outgrowth is called as aril. The edible flesh of the lychee fruit are highly developed arils surrounding the seed. Funicle, hilum, micropyle transformed into seed placenta, hilum, seed pore. During transformation after fertilization, the other parts of flower, for example, sepals, petals, stamens, stigma, style, are dried up. Ovary of the carpel by the influence of hormones transformed into fruit. Antipodal synergies are damaged. Nucellus become perisperm or damaged. Ovule pore becomes seed pore. Significance or importance of fertilization. As a result of fertilization, the ovule which are located inside the ovary transformed into seed and the ovary transformed into fruit. Seed is the main source of new progeny. If seeds would not have formed, most of the flowering plants would have been extinct. Not also that all the animals, including human, are completely dependent on the plant for food. It restores the diploid condition by fusion of haploid male and female gamete. It results in the formation of triploid primary endosperm nucleus which develops into endosperm in the seed. It provides nourishment to the developing embryo. It brings about recombination of characters resulting in variation among the offspring. Okay, your homework: definition of fertilization, triple fusion, double fertilization, synergids, syngamy, antipodal cell, egg apparatus, and one difference between fertilization and double fertilization. Number 3, draw a labeled diagram of fertilization process. And number 4, describe the fertilization process.
Describe the male and female development process.
For objective, you may follow your textbook, you may follow board questions and then solve your test paper. Thank you to stay with us.